In this module, we will discuss that how hybridomas for antibody production are made and how they are isolated and how they are stored. Now, the cell growth and storage that is carried out for development of cell banks, as we have discussed in earlier modules, that we need cells, their storage, and that is carried out for the development of the cell bank. We make the cell bank where we preserve the cells. Now, hybridomas, they are primarily grown for their products, that is, hybridomas, they make their product is monoclonal antibodies. So it means that we have to store the hybridomas in sufficient quantities so that we can produce the desired antibodies. Now, in this respect, what we do, that the simplest method for antibody production is a static bulk culture of cells which are growing in tea flasks. That the cell line culture, they are grown in the static, that is, the flask, they are not shaken or stirred, they are left as such at required temperature, at required condition in the tea flask. Now, the tea flasks, they are the flasks which are designed for tissue culture and have various media capacities and cell culture surface area. That is, their surface area is sufficiently large so that the cell can be cultured on them. Now, these cells from a working cell bank, so we get the cells from a working cell bank vial. That vial is thawed, that is melted, and it is thawed rapidly into 1.5 millimeter medium containing 10% fetal bovine serum. That is the recipe. That is the 1.5 mil of that in the medium. We take the vial and put those cells into that vial. Then the vial is placed in an incubator at 37 degrees centigrade, and this incubator is supplemented with 5% carbon dioxide. That the 5% carbon dioxide is just flushed into this incubator continuously. As the cell division starts in the tea flask, the flask size is increased using the medium supplemented with 5% again fetal bovine serum until the desired volume is reached. Desired volume means we keep on putting that fetal serum again, time and again, as soon as it is used by the cells so that we get the required amount. As we get the cells grown in tea flask, sufficient amount, then the cells are left to divide until all nutrients, they are utilized and cell deaths begin to occur. Naturally, when there is no nutrient, the cell death will occur. So, then, as we get those cells, the monoclonal antibodies, they can also be produced in ascitic fluid in the mice. You know, we have described that the ascitic fluid is the one which contains a high concentration of immune or antibodies which are present in the peritoneum cavity, which are produced in the peritoneum cavity of the ascitic fluid. Now, hybridoma cells, they are injected into the peritoneum of the mice and allowed to grow there. So this is another method where, as a matter of fact, the one is we take the vial from the cell bank and grow the cells in the T flask. Another is we can grow, or we can take the antibodies from the ascitic fluid, and for that hybridoma cells, they are injected into the peritoneum of the mice and allowed to grow there. These cells are secretory and produce high levels of monoclonal antibody in the ascitic fluid. So as they go into the cavity, they are secretive, they produce high level of monoclonal antibodies in the ascitic fluid, and the number of in vitro bioreactor system. Now, this is another system that we can also use a bioreactor rather than using the mice or these animals. We can produce the monoclonal antibody through the bioreactor, and that these methods are in vitro bioreactor system, and they have been developed to produce high yield of monoclonal antibody in small volume of fluid. That this is a continuous in vitro bioreactor where you can get the high concentration of the monoclonal antibody in the fluid. Now, in this picture, the bioreactor for antibody production is being shown, where you have got a vessel which contains the medium, then there is a cell growth chamber, and they both are separated by a semi-permeable membrane. Now, in that case, what happened? That in the semi-permeable membrane, the media that can creep in, but the cells, they can utilize that and grow and make the cells more and more, and then we can take out these cells. Now, further, what happened? That you can purify these antibody molecules or hybridomas through the fine capillaries which are there, and tangential flow, it is in that bioreactor where the cells are separated and cells are cleaned, and they are hollow fiber media, that is the fiber, they are hollow and the cells are attached to them, and the media, it is cross-flowing and making the cells grow again more and more. So, the culture medium that can be changed to maximum cell growth and health. So, you see that here, rather than the mice and other T flasks, the bioreactor has got a system where they are separated by a semi-permeable membrane, the culture medium that can be changed, and as the culture medium is changed, the metabolites of the cells and all these, they are also excreted, and the cell gets a good, healthy growth. Then the fluid can be removed from around the cell to harvest the antibody. As it is in the second chamber, there is a port from where you can remove the cells to harvest the antibody, remove the fluid there. Now, these bioreactors, they are the system, do produce high yields of antibody, but can be problematical to set up and run, because in that bioreactor, you have to keep the sterilization more and more and keep the condition more sufficient. Now, cells are grown in the bioreactor for up to 6 weeks. So then, that the colon that is used must be stable. That is another drawback, that the clone which you use must be stable. That is, since it is the 6 weeks, and the antibody which is produced in high concentration without the presence of medium component, that is the advantage of this, and thus make it easily purification, and we get a better product.